Okay, so I think we'll go ahead and get started. So I'm really delighted to have this opportunity to introduce Jason Ernst as our UCLA COVID-19 seminar series speaker for today. Jason is an associate professor of biological chemistry, computer science, and computational medicine here at UCLA. He obtained his Bachelor's of Science in Mathematics and Computer Science with high honors from the University of Maryland, College Park, followed by a PhD in Machine Learning from Carnegie Mellon University. And he was then a postdoc at MIT under Professor Manolis Kellis, where he worked in computer science and artificial intelligence. We were really lucky to recruit him to UCLA in 2012, and he's risen rapidly through the ranks here. He has a very impressive CV and he's really won numerous awards. But what was most striking to me is the number of high impact papers he's generated that have greatly advanced the field, including several Nature and Nature Biotechnology papers, which were each selected as one of the top 10 most influential papers of the year in regulatory and systems genomics. Now, since the COVID-19 pandemic hit, he's turned his computational approaches that utilize machine learning to analyze high-throughput data to the SARS-CoV-2 genome. And we're very excited to hear what he's going to tell us about today. And his talk is titled Single-nucleotide conservation state annotation of the SARS-CoV-2 genome. So Jason, please go ahead. Take it away. Sorry, I should also just, before you get started, remind everybody to please turn off their audio, so mute yourself and turn off your video. And if you don't, then I'll go ahead and do that so that we don't disturb our speaker. And if you can hold questions probably to the end, although if there are some burning questions that are not too disruptive, we can try and take them in the middle of the talk. Thank you.
And you can see the screen?
Yes.
Okay. So, thank you, Bridget, for the introduction, and thank you everyone for attending. So I'm going to be presenting some work that's been done by my PhD student, Subin, who's on the call also, about single nucleotide conservation state annotation of the SARS-CoV-2 genome. So what you see on this slide is the reference genome for SARS-CoV-2. And it's sort of key importance to understand this genome, and in particular also the mutations that are occurring. So for those of you that were on the seminar last week, you heard Leonid Krukliak's presentation on the analysis of the mutations locally in the Los Angeles area. And what's happening with the genome is it's accumulating mutations, and it's important to understand the genome sort of at the level of single nucleotides. For example, these mutations could, in principle, have implications to the effectiveness of therapeutics and vaccines if the genome mutates in certain ways that changes its behavior. So that motivates this need to sort of in more detail understand the genome and potential mutations. So one potential way to sort of do this, or to start getting a better understanding, is to try to develop more systematic annotations of every base in the SARS-CoV-2 genome. So this is something that my group in general, we do a lot of work trying to think about ways to better annotate the genomes focused in the human genome, so we don't specifically focus on virus genomes, but we've tried to apply some of the tools that we've developed previously in this context. In particular, we're going to focus on comparative genomic strategies for annotating genomes. So the idea with comparative genomics is you have a genome that you're interested in understanding, in this case, the SARS-CoV-2 genome, and you compare that sequence to the sequences of a number of other genomes. So in this case, I'm showing you an alignment of sequences of SARS-CoV-2 and then 43 other genomes, including SARS-CoV, and then 42 other sarbecovirus genomes that infect bats. And what you see on the right is the phylogenetic tree of these genomes. And just to sort of make the distinction on the other slide, the phylogenetic tree was of different strains within this outbreak. Here we're comparing other viruses that aren't SARS-CoV-2. And the question is sort of how can we use this information to systematically annotate every nucleotide in the genome? So there's a lot of different uses of these types of alignments, and a number of people focus on specific evolutionary questions, or they have specific directed tasks. But what we're interested in is using this information in this alignment to say something about every single base in the genome. And currently, I mean, sort of a predominant strategy is you give a score to each base, a constraint score. And there's methods such as PhyloP and FastCons, which have been around since about 2005, where people have a phylogenetic model that measures constraint. And so this is showing you a view from the UCSC genome browser, where they have a pipeline after they produce one of these alignments that they also score each base based on these two different constraint scores. But I mean, one thing which you might notice is that there's a lot of information in this alignment that sort of gets lost when you try to compress it all into a single univariate score. So this is just giving you an example where you can see two different columns where they have both limited, very low scores, but in a sense they're different, where in this case, this position only aligns to one other genome, and then the dash means indels, while over at this column, there's an alignment to many other genomes. It's just not matching. And when we try to compress all this information to a single univariate score, that information is getting lost. So that sort of motivates the question, is there a way that we can effectively summarize the information in the alignment while preserving additional information and use that to annotate each base of the SARS-CoV-2 genome? So previously, my group has developed an approach, and it's called ConsHMM, and we developed this in the context of annotating the human genome. So in the first part of the talk, I'm going to explain this approach and also give some background on what motivated this approach. And then I'm going to talk about our specific application to applying ConsHMM to the SARS-CoV-2 genome. And then in the last part, I'll talk a little more in depth about relating these annotations to within the pandemic mutations. So this first part is work done by a former PhD student in my group, Adriana Arneson, and this methodology is published. So what I'm showing you here first to give some background is what motivated this methodology, ConsHMM, and it was inspired by previous work that I had started during my postdoc on ChromHMM, which was a method to annotate genomes de novo based on epigenomic data. And this methodology has sort of become widely used in the biomedical research community for very diverse sets of applications. And so I want to give a little background on that for those who aren't familiar with this. And then the methodology that I'm going to talk about in the context of comparative genomics is an extension on this. So it's sort of unique in the comparative genomics sense, but coming from the success it's had in the epigenomic space, we felt that a lot of the ideas could also have value in annotating genomes based on comparative genomics data. So the idea with the methodology when we're annotating genomes based on epigenomic data is that we can have different types of chromatin marks, in particular modifications to histone proteins that occur in different combinatorial and spatial patterns. So we might see pairs of different markers on top of the genome co-occurring together. And then they might also occur in like spatial domains. So then the question is, computationally, can we identify these patterns of sort of co-occurrence or spatial patterns? And then once we discover these patterns, then we can go across the genome and annotate each location to an instance of one of these patterns. So then this becomes a strategy to de novo annotate genomes. So what you're now seeing here is actual experimental data. So the rows correspond to different tracks. And then what we do is we find these patterns of different marks, and we discover the patterns, and then we can annotate the genome based on them. So you saw like a red annotation and this green annotation, and they can correspond to sort of probabilities of having different marks together. So it's in a probabilistic sense. So the rows here correspond to different states. The columns are these different markers. And then once we have these annotations, we can go across the genome and then, have these state annotations and then compare those to other types of annotations and compute various statistics and start giving biological interpretations, such as something's a promoter, enhancer, transcribed, repressed, or heterochromatic region. So this strategy has now been used to annotate a wide variety of different cell types. And I just wanted to highlight one location. So I worked previously in part of the Roadmap Epigenomics Consortium, and we annotated about 100 primary cell and tissue types. And I'm highlighting one gene here that's sort of important on the host side to SARS-CoV-2 entry. And you can see that it has different types of chromatin state annotations in different cell and tissue types. So certain, for example, gastro-related cell or tissue types, you might be able to find enhancers in certain regions and so forth. So this is a resource for those also who are studying SARS-CoV-2 from the host perspective. But my reason for sort of showing this is to motivate the methodology that I'm now going to present, where we're focusing on annotating the viral genome. So what we developed here is a methodology, ConsHMM, where it's a similar idea where we want to learn different states of the genome de novo, but instead of having different tracks correspond to different chromatin marks, the different tracks correspond to different species. And then instead of having an epigenetic mark present, what we do is we have an observation, one of three possible observations for each species. It either aligns to and matches the reference genome. In this case, it's a human reference genome. And what I'll show you later will be the SARS-CoV-2 genome. It aligns, has a nucleotide there, and doesn't match. So that's X observation. Check is the aligns and matches, or there's no nucleotide there present in alignment, which can correspond to an indel or a gap. And we do this modeling making very few assumptions. We don't use a phylogenetic model at all. The observations are modeled with the product of independent multinomial random variables. And we do this annotation at single nucleotide resolution, which in contrast to what I showed with ChromHMM, that was at 200 base pair resolution. So this is now to illustrate the ConsHMM modeling. So what you see here is the reference DNA that you're trying to annotate. And what's shown below this are hidden states that are inferred. And over here are the observations, which corresponds to, in this alignment, did that species align and match the reference genome? Did it align but not match? Or was there no nucleotide there? And then in different states, we have different probabilities of these different observations for different species. So then we can associate different columns with these different states. So in the state 2, we might have higher probabilities for other observations, and then those locations can get assigned to those states. And then there's also behind the scenes some information on the relative likelihood if you're in one state in the neighboring position, you'll be in the next state to capture some of the spatial properties. So then just to briefly show you some results when we applied this to human, and then I'll get into the SARS-CoV-2 annotation. What you're seeing, the rows correspond to different states, and then the columns are individual species. The left-hand side is aligning human, so that's the check or X observation. And then the right-hand side is you align and match human. So you have a nucleotide that's the same as human. And what you can see here, for example, is there's one state at the top here, which corresponds to you align and match all these different vertebrates. So this is based on a 100-way alignment all the way through fish. So we have some positions in the genome which align and match all the way through to fish, while others do sort of subsets of different species. And there's one unique state down here which sort of you have alignment and matching for some distal vertebrates, but not closer mammals. And this can be related to pseudogenes and alignment artifacts, but it's showing that this, like the traditional constraint scores here, the phylo P and FASTCons, are also giving this a high score. And down here are the different alignment patterns corresponding to these two different types of states. But by having the state information, we're able to give a more detailed view of the alignment patterns and separate out what might otherwise get sort of be viewed as constrained by a single univariate score. And this is showing you some additional states as well, the enrichment relative to protein coding genes. And what you can see is that this type of annotations can de novo pick up like single base resolution patterns. So we're seeing this 3 base period oscillation corresponding to the synonymous, the third codon position. And then it is also capturing constraint related to canonical splice sites. And then we can see an inverse pattern for positions that are in the third position. And these states correspond to when you have deeper alignment, but not as deep matching. And then there's other states where you don't have strong aligning or matching at all. And then the last example here, I'm showing you around transcription start sites. So it was able to find a state that was on the order of 30-fold enriched for transcription start sites, protein coding genes, and then 2 states here that have a high enrichment around transcription start sites of pseudogenes. And the transcription start sites state has sort of a pattern where you have many species aligning and matching, but not with particularly high probability. And these 2 around pseudogenes have this sort of pattern. You have these distal species aligning, but not these closer ones. So the point of this is by looking at these sort of more detailed alignment patterns, we're able to de novo sort of find patterns in the alignment and then annotate each nucleotide of the genome based on these patterns. So then my student Subin tried to apply this approach now to annotate the SARS-CoV-2 genome. And we actually use 2 different alignments. They're both from the UCSC genome browser, and I'll talk about both of them. The first one is a 44-way alignment of sarbecoviruses. So this is sort of relatively close to SARS-CoV-2. And then they were all with bat hosts, except for SARS-CoV-2 and the original SARS-CoV. And then the next alignment I'll talk about afterwards is a 56-way alignment of more general coronaviruses. So this is sort of going more distal evolutionarily and not sort of restricted to bat hosts where they've included any vertebrate hosts. And sort of you can see the furthest one out over here in this alignment we're going to consider is just this one over here, sort of the 4th one other than SARS-CoV-2. So first to talk about this one about the bat alignment here. And just to illustrate, again, sort of what we're doing, the input here is this alignment. And then we've encoded it with these, is it matching, aligning but not matching or not aligning here. And when you see the period in the browser, that means there's matching. When you see the dash, that means it's sort of an indel or not aligning. And then when you see a different letter here, that means it aligns, but it's not matching. And then what you see below this is the output of the method of at a specific position. So what we're doing is we're now assigning each nucleotide to a different state. In this case, we have 30 different states, and I'll show you what these states correspond to. But it's what you can see, for example, this where we have these yellow columns, all those positions got assigned to some state which is associated with high sort of alignment and matching, because every species is sort of, or almost every species is matching in these columns. So what you see now is the actual states of this model, and we've clustered the columns based on the values you're seeing. And the left-hand side corresponds to these aligned probabilities. So those are the check or X observations. And then the right-hand side is the check observation. And then I have the individual other virus genomes labeled on the bottom. The far left column is a heat map of the coverage. And here you can see the probability heat map on the right to show sort of red means a higher probability, blue means lower. And then to highlight some specific states, and what I've done on the right here is I've shown some specific enrichments for these different states. So we've gone across the genome, annotated each nucleotide to one of these states, and then we can see of the different genes of SARS-CoV-2, and then also regions of interest. So for example, the part that binds ACE2 or the receptor binding domain. So these are all uniprot annotations. What's their enrichment? And then on the bottom, it's showing what percentage of the genome that corresponds to that column. And we've computed a number of other enrichments that I won't have time to go through all of them, but just to highlight an example here. So, I mean, the first state, when you have this high aligning and matching through all the sarbecovirus genomes, that is actually still about half the SARS-CoV-2 genome. But then we can contrast it here at this other extreme where we have limited alignment to any other sarbecovirus except for RATG13. And this one is assigned to about 1% of the genome. And that's where you get this high enrichment for ACE2 binding. So it's a 22-fold enrichment. And then down here, we sort of have this unusual pattern of only a subset of specific species align, and then some other ones are missing. A few are missing down here. And this one has both a strong enrichment for intergenic bases. So about 2% of the genome is not assigned to a gene. And then there's, or an open reading frame. And then there's also another ORF10 has a high enrichment for this state. And this ORF10, there's been sort of work in the literature trying to figure out if it was protein coding or not. And there's sort of evidence that it's likely not protein coding, it's not transcribed. And then there's other comparative genomic work based on codon signature, which is also saying it doesn't look like it's coding. But you can see how it's sort of, we're able to de novo discover these specific combinations that are corresponding to these other genomic features that the model didn't have any knowledge from beforehand. And then to now go through this other alignment. So in this case, we're working with this deeper set of coronaviruses. So what you're seeing now is, it's similar before. The left-hand side is this align or match the reference, and then the right-hand side is you match. So that's the check observation. And then in this case, I've color-coded on the top what host species it is, whether it's bat, bird, human pangolin, or another vertebrate. And sort of one thing to sort of highlight here is which columns correspond to viruses that infect or known to infect humans. So we have SARS-CoV, there's MERS, and then we have 4 different common cold viruses. And these common cold viruses are interesting, and right now there's a lot of discussion about why different individuals respond differently to coronavirus. And I mean, there's probably a lot going on, but one thing that's been discussed is prior infections to common colds could cause the immune system to behave differently. And then there's been recent papers showing that people who haven't been exposed to SARS-CoV-2 still have epitopes targeting it, and some of it could be from prior infections to these common cold viruses. And you can see like the specific states here, which you have sort of this agreement with the common cold. So there's certain places where all the coronaviruses share, but then there's also some states which are shared by the certain common cold viruses. So some, all of them, and then there's some other states, it's depending on which common cold virus you're looking at. So this could potentially be a resource for sort of teasing out some of those differences. And then to make the point here that because we're working with this sort of deeper evolutionary set of genomes, in this case, the state with this high aligning and matching throughout all the genomes now only covers 9% of the SARS-CoV-2 genome. And then what's interesting up here, so there's a state that is a little in between the lines, but the state here where you have the bat coronavirus and the RATG13, and this pangolin coronavirus has high aligning, but then not these other two sarbecoviruses. And this corresponds to regions of the ACE2 binding in the receptor binding domain, while other parts of the genome, this pangolin coronavirus has lower aligning. And this has been discussed before, of this higher agreement for this pangolin coronavirus in this sort of key region. And so this is a type of feature which is sort of automatically coming out of these different states that are learned de novo. And now in the last part of the talk, we'll talk about some of the analysis we've been doing using these annotations to analyze the pandemic mutations. So what we did here was we took the mutations that were available in next strain in May of 2020, and we identified a set of 1,410 positions out of about the 30,000 positions in the genome, where there was mutations that were observed in at least 2 sequences or samples. So these are non-singleton mutations. And then we computed state enrichments for these mutations. So where are these mutations falling? Are these positions where we've observed a recurrence of a mutation in more than one sample? So we saw, I'm showing you, more to the right is enrichment, to the left is depletion. So that's on a log-based 2-fold scale. And then I put a star if this was statistically significant. And what you can see here is the more conserved states, such as the state here, which has the high aligning and matching, depletes for these mutations, and as well as some of these others that also sort of align pretty deeply. So we're looking at the 56-way one with high matching. And then those that are enriching, so there's this state which has sort of a deep alignment, but very little matching, and then there's other states over here which also have sort of not as deep aligning, but then also not deep matching as well. So one of the points of this, and if you're sort of familiar with also what's seen in humans, is that places where you have more constraint based on comparing cross species, you're also less likely to have variation within the species. So we're seeing this depletion of mutations in a sort of in these states that we have more evidence in a sense based on the comparative genomics that they're potentially more sort of important potentially to the species, and that they need to, or the virus, and they need to maintain that. But then when you actually do see a mutation in one of those states, that could potentially be a more consequential mutation. And then going to the 44 way alignment, we see again the state associated with aligning and matching through all the bat coronaviruses, showing a depletion of these bases with recurring mutations. And then we had several where it was significantly enriched as well. And then we've done, so this is a little more technical, but we've tried to do quantitative evaluations of how much information is there in these annotations to predict these bases with these non-singleton mutations. So these are the negatives in these analyses versus bases without. So the bases without have either no mutations or only a singleton mutation. And we've also matched this based on the nucleotide. So there's a different mutation rate depending on the nucleotide and the reference. So we controlled for that and then tried to see how well these different conservation state annotations can predict these bases. In a sense, the way to think about it is in aggregate, potentially these positive bases, so these bases without the non-singleton mutations are sort of more functionally important overall versus those that have these recurring mutations. And what we can see is there's some predictability. There's, on the x-axis, you're seeing the false positive rate, the y-axis, the true positive rate for an ROC curve. And we're comparing the two different alignments, the ConsHMM annotations, and then a simple combination of them where we take whichever state sort of has a higher enrichment. And this was evaluated on a 10-fold cross-validation. And then we compared to sort of another like more standard way people talk about different types of mutations, whether it's intergenic, a synonymous mutation, a missense, or a nonsense mutation, as well as also like what gene the mutation is in. And what we found is it was significantly more predictive of the bases that are, you're gonna see these recurring mutations versus not when we used the conservation state annotations versus when we used the gene annotations or the mutation type. And then when we compare it to the constraint scores, in this case, we saw a slight advantage to the conservation states, but it wasn't statistically significant. But there's sort of other advantages of the conservation states for other types of analyses. As you saw before, it's sort of very multi-dimensional, which you lose with the constraint scores. And then to just sort of give specific examples of mutations. So what we did was we took the mutations from this specific paper. So they defined mutation hotspots. They found a set of 13 different mutations in SARS-CoV-2 at the time, which they considered hotspot, which I think was found at least 10 times. And then they sort of broke it down into those that were found sort of only in Europe and North America, or then there were some that were found both in Asia and Oceania and North America, and then the time period. So the ones that were found in Europe sort of evolved later. And what I've highlighted is in purple are those that were found in Europe and North America specifically, and then the rest are those that were shared. And one of these is sort of this sort of well-known mutation in the spike protein, D614G, which has gotten a lot of popular press and was also discussed in the seminar series last week. And one thing which we sort of noticed was that of the 8 that were specific to Europe, 6 of the 8 were in the state which was aligning and matching through all the different SARS-CoV-2 So even though state 17 I was showing you before is depleted in general for mutations, in this case it had slightly more than expected, and then those that were shared, actually only one of the 5 had this specific state. So, I mean, it's small numbers here, but it was sort of close to statistical significance, this difference. And then just to highlight one specific mutation here that was notable, it was found in this state here, which was lining and matching through all the coronaviruses. So this was sort of generally preserved evolutionarily, positions assigned to the state, but there's been one hotspot mutation where, this is an NSP13 helicase where it's now been found in a number of sequenced viruses. And then in order to sort of convey some of this mutation information in a more accessible form, what we did was we created a track which is showing a score of the depletion level. So if it's higher, that means it's the position at that base. That base is assigned to a conservation state that is depleted. And then if it's below the line, it's assigned to a conservation state that is enriched. And we only use those conservation states that were showing significant depletions or enrichments in one of the two models. So these are the state annotations from the two models that we used, and we've now converted this also back to score track. I mean, now it's sort of similar to the constraint scores, but it's directly based on these conservation signatures and the mutation enrichment and depletions as well. So the intuition here is that these states that are, again, sort of depleted in general for mutations, when you do observe them, it might be of potential like more interest. But again, it's just sort of one signal in the analysis. So to summarize, what I presented to you was a method, ConsHMM, that can learn conservation states from multiple sequence alignments, and it's analogous to ChromHMM for chromatin state annotations. And we applied ConsHMM to annotate the SARS-CoV-2 genome based on two different multi-genome alignments. And then we conducted mutation analysis and identified specific states showing enrichments and depletions for mutations. And then I'd like to acknowledge sort of two people involved in this work. So the ConsHMM method development was done by my former PhD student, Adriana Ernst. And then the SARS-CoV-2 work was all done by another bioinformatics PhD student, Steuben Kwan. And also like to acknowledge Sriram for some comments on the work. And then NextStrain provided the mutation data we used, and UCSC Genome Browser provided the different virus alignments, and as well as the funding for this work. And then I leave up here certain resources we've made available. So we have the conservation state annotations, both in a browser view, but also in bed files if you want to do your own script analysis on it. And then those mutation depletion tracks. We have a preprint on the work, and then also links, more generally, if you're interested in the ConsHMM methodology, or also the ChromHMM method, and then there's also some pointers to some of the annotations that have already been defined for human and other species. And if you have questions about that, you can contact me, and I can take questions now. So thanks for your attention.
Great, thank you so much, Jason, for a wonderful talk. So if people would like to ask questions, you can raise your hand, or you can type in the chat, or you can turn on your video and audio to ask questions. Doug, go ahead.
Oh, okay. Jason, yeah, that was really interesting. I'm curious about, so SARS-CoV-2 is a single-stranded RNA virus, and one common application of phylogenetic comparison in RNA is to use it to define secondary structures to look for for nucleotides that co-vary. They're not constant, but they maintain base pairing within a structure. I was wondering if how that would look within your models. You know, would those, if you had a nucleotide on either side of a stem co-varying, would those come out as in the same state, or how would that look?
So I guess it, like if they're co-varying in the same way, so if they're both like present in the same species and absent in the same species, opposed to this one's present and the other one's not present.
Yeah, so you'd have an A over here and a U over here in one species, and then a G and a C in another species, and then a C and a G in a third species, and so on. You know, so you'd maintain base pairing across a stem. Nucleotides can change within.
Right, yeah. I don't think that would directly come out of this modeling. I mean, you would see, I mean, you would, if it was the case of like these subset of species all sort of still match this reference genome at these positions, and then these other subset of species no longer match the reference genome, that would fall into the same state. But there's some higher level sort of combinations that don't necessarily come out of this. And there's been other directed analyses in the RNA space.
Right, yeah.
Genome using the evolutionary information as well. So I didn't sort of go into a lot of other applications of these alignments sort of outside of trying.
Yeah, it might be interesting to look at the regions where there's known structure, the 5' and 3' end, and the pseudoknot in the frame shifting in the middle of ORF1. It would be interesting to look at.
Yeah, okay.
Great. I also just want to mention that the recording of this presentation and all the others from the seminar series are available through the DGSOM, and it takes a couple of days to about a week, but this recording will be up and available for everybody. I guess I have a question, but it's more of a clinically related question, Jason. So I'm thinking about how this data is going to help us think about why this virus seems to be more virulent, why it seems to have, why it seems to be maybe affecting more people than we'd imagined, and if there's any clue in there as to why it's affecting more people more than others. Is there something that is conserved or not conserved between, for example, MERS or SARS-CoV and this particular SARS-CoV-2 that can give us any kinds of clues?
Yeah, so there's been some work where they ask sort of more directly, and you can actually, let me share the screen again, sort of see this. So the question would be like, if you have the genomes that infect human, these cold viruses, but then, and then you have other places where you have SARS-CoV and MERS. So there's certain states, for example, where you have some matching to SARS-CoV and matching to MERS, but then none of the cold viruses match. So these types of bases are good candidates for potentially like sequences that could cause things to have a more severe outcome in human. I mean, in general, this analysis, it's a little tricky because a lot of, I mean, just SARS-CoV, it's closer evolutionarily, and then the common cold are much further. But there's sort of some signal because MERS is somewhere in between. So there's been, at least there's one paper in PNAS where they did analysis specifically targeted at that question of discriminating sort of the comparative genomic signal of SARS, SARS-CoV, and MERS from common cold. But you can sort of see de novo from these states where we have those higher probabilities in SARS-CoV and MERS, but not the common cold viruses as well. And then, yeah.
So can you use these data to drill down into specific genes that you think might be different, or is it just too difficult to compare?
I mean, so like if we took these specific states, we could go and see like in what genes of the genome they're in, for example. And then for this analysis, it might also make sense to run a model specifically targeted on these other the subset of genomes, which we haven't done yet.
Good. And is that a direction you guys are going in?
Yeah, it's something I've been thinking about.
Okay, great. Thank you. That's great. Are there any more questions? Okay, great. Well, if not, I'm just going to say thanks again, Jason, for a really, really interesting talk. And again, we'll put this up on the website. And yeah, we'll see everybody again next week at noon Wednesday for the next seminar series. So thank you everybody for joining us today. Thanks. Bye.